Hello, my name is Vuong Tran. It's my honor to present the summary of our study on behalf of my co-authors. In this study, we performed bioinformatic analysis on KIT expression in asthmatic airways and reported a link between KIT expression, mast cell abundance and activity with Th2 high endotype. In this study, we selected 5 gene expression datasets, including bronchial epithelial brushing samples from healthy controls and patients with asthma. By gene expression analysis, we found that KIT expression were higher in asthma. 2 different methods of in silico cytometry show that the abundance of mast cells and Th2 cells were higher in patients with asthma. Focusing on asthma, we included 2 other gene expression datasets of patients with asthma only. We grouped asthma patients into KIT low or KIT high based on the expression of KIT, and we found that the KIT high group has a higher abundance of mast cells and basophils. In agreement, pathway analysis showed a higher activity of F6 receptor 1 signaling. We then cluster asthma patients into Th2 high or Th2 low based on the expression of CLCA1, both TN and SERPIN-P2 genes. And we found that the KIT high group included predominantly Th2 high endotype individuals. Notably, because most patients with asthma in the included datasets were taking asthma drugs, our findings indicate that KIT expression, mast cell abundance and activity remain elevated in asthmatic airways regardless of treatment. In summary, by achieving consistency across multiple datasets, our results reaffirm and re-emphasize the importance of KIT or mast cells as therapeutic targets in asthma. Thank you so much for your attention.